all, good afternoon. This is chapter 8. I'm still Dr. Beal, and I want to get through this idea of what happens when there's a mutation or a change on the chromosome. So we have been looking at some variations that cause phenotypes to ultimately be different. A lot of what we've been looking at has been recombination. Now I want to look at what happens if it's something really changes. What if my chromosome is too long? What if it's too short? What if I'm missing a piece? What if non-homologs cross over during meiosis? And I have a whole bunch of combinations for that. Before you watch these, you should definitely be comfortable with the idea of a homologous chromosome. Review what diploid and haploid really mean from the very first day of class. The difference between meiosis 1 and meiosis 2. What separates, when does ploidy change, when do we really go through the process of independent assortment. Those things are going to help you as we go forward. And finally, just take a quick look. We had in our lecture on chromosome structure the concept that genes were on the chromosomes. I want you to think back too to Bio 1. Not only are genes on chromosomes, but those genes are made out of code of DNA, so A's and C's and T's and G's. What happens when we mess with that? For the first video, I'm just going to do these first two. I'm going to talk about some various words and vocabulary you need for chromosome numbers, how to talk about whether you have 2 copies of everything or just 1 extra copy of a chromosome. And then we're going to look at non-disjunction, which is when meiosis does not separate during anaphase 1 or anaphase 2 correctly, and how that results in having too many chromosomes or an aneuploidy. So aneuploidy. Aneuploidy is having too few or too many chromosomes of one. Most people at all of their chromosome sets, their homologous pairs, have a disomy. That means you got 1 from mom, 1 from dad. You have 2 of every chromosome numbers. And we've been talking about that being diploid, but it's really having 2 of each. A trisomy, something where we talked about Klinefelter syndrome in the last couple videos. This is where you have 1 too many chromosomes. A monosomy, like Turner syndrome, is where you have 1 too few. In humans, if you have a disomy, you have 46 chromosomes. If you have a trisomy, you have 47. You have the normal number of autosomes plus 1 extra sex chromosome if you have Klinefelter syndrome. I'm going to give you some other examples in this lecture. And then monosomy, the only one I actually know of in humans is Turner syndrome, where you're missing that second sex chromosome. You would only have 45 chromosomes. And we need to talk about these 2 ideas here too. We've said them, but I don't think I've really detailed them yet. Disjunction is during anaphase, when you go from metaphase to anaphase, whether it is just metaphase to anaphase, metaphase 1 to anaphase 1, or metaphase 2 to anaphase 2. It's when you go through normal separation of the chromosomes, either homologs or sisters. Non-disjunction is where something is wrong. This is the mistake. Let's write mistake here. It means that the chromosomes fail to separate when they were supposed to. In this image here, the first meiosis 1, first part of meiosis 1, the homologs fail to go through disjunction. They end up doing it wrong. They go through non-disjunction. And so for meiosis 1, these cells are empty, having no member of this chromosome, and these still have homologous pairs. This is going to result in gametes at the end, where I have gametes with just 1 too many chromosomes in them. They're trisomy. And I have 2 gametes with only 1 chromosome, or 1 too few. If non-disjunction occurs during meiosis 2, here it's occurring over here, where we're separating sister chromatids, I get 2 normal gametes, and then I get 1 gamete that has a trisomy, 1 too many, and 1 gamete that has 1 too few. And they pass on to individuals when they go through fertilization. Euploidy, this EU, means true or accurate. If you remember, you're a eukaryote. Karya is nucleus. You're a true nucleus-bearing organism. Euploidy is the true number of chromosomes. Aneuploidy is having not the true number of chromosomes. Diploid is 2N, triploid is 3N, and then we have polyploidies that I'm going to talk about today. That's where you have 3 sets, 4 sets, 5 sets, and etc., even more, of entire chromosomes. I'm going to go back here and separate these words from each other. A disomy is referring to having each chromosome is paired, one from mom, one from dad. Diploidy is having a full set from mom and a full set from dad. Triploidy implies there's a third parent. Not really, but if you're thinking about it for the first time, if you are polyploid, you could possibly, if you're a plant, have 5 different parents that have contributed one set of chromosomes to you. It doesn't occur in animals. So I have a question for you. This is not in your handout, so I took it out. But just for you to think about, a human female with Turner syndrome, and so you have to think about what Turner syndrome was, expresses an X-linked trait, hemophilia, and her father did too. Who underwent non-disjunction during meiosis? If she expresses hemophilia and she has Turner syndrome, where did she get her single X from? Well, she must have gotten her X from her father. Whoop, I just lost my thing. She must have gotten her X from her father with hemophilia on it because he only had one X to give. He's going to give his X. Mom, it doesn't say whether she had hemophilia or not. Let's say she did not. Mom is not a carrier, nor does she have hemophilia. She should have given a non-hemophilia, a completely normal X, but because she went through non-disjunction, my X makes it confusing, she didn't pass on anything, and her daughter ended up with her father's X and nothing from her mom. It's very likely in this case that the mom underwent meiosis incorrectly, and probably early on during meiosis 1, where her 2 X's all ended up in the same haploid cell together. So I want to zoom in on these idea of monosomies and trisomies and give you some more examples, because really at this point all I've given you is examples with the sex chromosomes. And when you have a monosomy of the X, the Barr body really makes that less easy to see because everyone only expresses one X. So having a monosomy or the loss of that one X chromosome is less, devastating or noticeable than if you have a monosomy in a somatic chromosome in one of your autosomes. Normally what happens if it's not a sex chromosome is you really only have one copy of each gene, which means for that chromosome you're functionally haploid. If you are carrying a mistake, if you have one bad gene on your entire single chromosome there that is not going to allow you to develop, you don't have any backups. Monosomy basically unmasks, and I want to come up because this word was confusing to people on the exam. Mask means to hide. Like wearing a mask hides your face. Unmask means to reveal. So you reveal it. If you don't have something that is hiding your recessive lethal allele, it expresses. Now what we also see, monosomatic gametes who are autosomally monosomatic. So does that make sense? They have their autosomes. One of them only has one. Okay. When you see that tends to have interference in the gamete development completely, and we find out that those individuals are generally not viable. We don't see monosomies of the autosomes in humans. What we think is coming back to is the idea in many cases that you actually need 2 copies of every gene, whether they work or not. You actually need that backup gene, and missing some of it makes you sort of haploid insufficient. Of haploinsufficiency, or you can't develop because you don't have your backup copy, even if it doesn't work the way that the body expects. Whereas a monosomy is the loss of one chromosome, a trisomy is the gain of one extra chromosome. We've looked at Klinefelter syndrome, where individuals had XXY. They had one extra sex chromosome. I also want to look at probably what is more familiar to you, a couple examples here of trisomies of the autosomes. This is an example of a trisomy at the 21st position, Down syndrome. Down syndrome is a 21st, one extra chromosome, and this tiny little chromosome here, this causes a whole host of developmental and physiological changes. People with Down syndromes tend to develop problems with swallowing as they're very little. They're very hard to nurse. They have problems with heart valve development. With modern medicine, we've been able to address a massive amount of the problems that are caused by this extra chromosome, whereas the lifespan of people with Down syndrome in the 1950s was into the mid-early 30s. Today, it's a completely normal human lifespan because we can fix some of the heart and developmental problems early on, and there's a lot of feeding strategies to help a baby better feed. I do want to point out that plants in general don't have the same kind of problems that animals do with increased chromosome numbers, including even being polyploid and making sure that you have whole other sets of chromosomes. This is Jimson weed. I would call this the devil's weed, just locally, so I think that might be what we locally call it. This has many trisomies when we look at the genetics of it. So I'll find trisomies of almost every kind of chromosome, and depending on which chromosome has three versions, I'll see the fruit looks different in this. Now, this is poisonous at all places. You can't eat this at all, and it hurts to touch it, and it stabs you. So I can't think of anything great about it, except that it's a really interesting example of trisomies in plants. I want to come back to Down syndrome just very briefly, and we'll come back and speak on it a little bit later in later lectures. The 21st chromosome has genes, and many chromosomes do, but when we look at the 21st chromosomes, part of the reason Down syndrome expresses the way it does is some of those genes on that 21st chromosome are dosage sensitive. Basically, the more I make of those genes, it changes my physiological and developmental responses. So this is basically like the more or the less of a protein I make because I have more or fewer copies of the chromosome, that actually affects the expression of various physiological states. This third chromosome, when you get the third 21st chromosome, we do know from years of research that this is very likely caused by non-disjunction in the mother as she's making eggs. It seems to increase with increasing age of the mother. We'll look later during gamete development, and I kind of hinted at it early on, when women make eggs, and those eggs are going to be the gamete that she passes on to the next generation. That process actually starts while she's in her mother's uterus during early development. The cells that are going to be the eggs are in prophase 1 before she is born, and they're already starting to go through synapsis and crossing over. So by the time you're 45 years old and you're wanting to have a child, your eggs have been going through synapsis for 46 years, possibly. And so it's very difficult for them to separate appropriately. It's part of why it's difficult to get pregnant as you age, among other things. But it's also why we see a rise in chromosomal number and abnormalities in egg production with advanced age. You're more likely to have an aneuploidy. Down syndrome is something that babies can survive with. So we see this big rise of aneuploidies. Aneuploidies at chromosome number 1 or 2 or 3 are generally not viable. The offspring won't develop at all. There is in your textbook a section underneath this part. It's in 8.2 about the ratio of age to aneuploidies. If it's your thing, or if you're considering that for your paper that you're writing for the class at the end, I think it's a really useful start to your search there. I want to introduce here something that I'm going to talk about at the end of video 2, actually, for this chapter, but it ties in with Down syndrome, so I want you to have it in both places. Familial Down syndrome, meaning it is passed through family lines, is not caused by an error in meiosis. It's actually caused by a translocation error. It's called the Robertsonian translocation. It's really well understood. I think in lab the other day I talked a little bit, before we did the DNA necklaces, I talked a little bit about the fact that we have this really famous translocation, which is the Philadelphia chromosome. This is another one that's really well known. A translocation is when chromosomes align, and they're supposed to align and form the tetrads with homologous pairs. If they align and form tetrads with non-homologous pairs, in this case chromosome 21 and chromosome 14 align, and then they form chiasmata and cross over, some of those pieces that were once on 21 get stuck onto 14. And that means that when the body is trying to make whatever it wants to make for chromosome 14, it's actually making the proteins that are on chromosome 21. When this happens and you're a carrier of this translocation error here, it means that when you make gametes, you are going to pass on one of a normal 21 and either a translocated 2114 or a normal 14. As that happens, it means that in many cases you pass on the gene right here, which because of dosage and the fact that making too much of it leads to Down syndrome, actually causes Down syndrome without a meiotic error. So this person inherits too much of this Down syndrome region, which causes Down syndrome in high dosages, and this person actually gets missing parts. They don't actually get enough chromosomes at all. They're monosomatic, so they don't develop. We'll look at this at the very end of video 2. If you're interested in Down syndrome as your presentation, sort of what I said before, this may be something to consider that is highlighting it in a way that you may be less familiar with. It may be more interesting to you. I have 2 other examples for you for trisomies. Trisomy 13 is Patau syndrome. Trisomy 18 is Edwards syndrome. Both of these cause early developmental disabilities. Patau syndrome has a whole host of problems. This is Patau here, and it's not labeled. Including cleft palate, which is what you're seeing in this baby, where the palate inside the mouth and around the jaw doesn't form properly. This is Edwards syndrome. This is actually formed by trisomy 18. So if someone would have an extra chromosome here. One of the things that's characteristic is this holding of the hand in infants. We can actually see the expression very early. Both of these result in developmental delays, learning disabilities, and other long-term developmental problems that are pretty devastating. Having an extra chromosome often means you're making too much of a protein. Generally, individuals born with both of these do not live a full lifespan. So let's take it one step more extreme. Let's talk about what happens if you got entire sets of chromosomes that you have too many of them, right? You got not just one extra chromosome, and we've seen one extra chromosome can cause a whole host of problems. What if I've got a whole extra set? Now, this does not occur in humans. I'm going to show you some plant examples because this is actually a really common characteristic in plants. This is table 8.1. I think you can fill out here. Aneuploidy is not the real number, or not the real number of sets of chromosomes. It's a chromosomal number abnormality. Monosomy is 2n minus 1. Disomy we talked about is 2n. Trisomy is 2n plus 1. It can be more, but not in humans. We don't see expressions of people with multiple numbers of chromosomes with one too many. True ploidy. This is having a multiple where they're divisible by the number of sets they have. So diploid, triploid, tetraploid, and so on. Diploid is what we have. We have one set from mom and one set from dad. Anything beyond diploid is called polyploidy. Poly means many sets. Ploid means sets. Triploidy, tri is 3, 3 sets. Tetra is 4. Penta is 5. And then I'll look at the very end of this video. We'll look at autopolyploidy and allopolyploidy. And we'll actually look at all of these. A lot of words that sound a lot the same. These are things in plants. Animals don't seem to be able to survive this condition, but plants seem to be primarily able to go through their evolutionary process by errors in entire sets of chromosome numbers. Autopolyploidy, and I'm going to do it, and I have it posted at the end, but I want to do a little bit of vocab reading with you. Auto means self. Poly means many. Ploidy means sets. Polyploidy is having too many sets of chromosomes. And I think the only place you've seen it so far is when you did the plant part of Gen Bio. We talk about the idea that the part around a seed that feeds the seed, the endosperm, is triploid. It's a result of a sperm fertilizing two egg nuclei, and it forms a blob that becomes the food source for the developing embryo seed in flowering plants. I want to introduce some actual other consequences of that. In autopolyploidy, that just says many sets. Either you make a gamete that is diploid, so you have a sperm that is 2N, it did not properly go through meiosis, or in the case of plants, you might have two sperm they're good looking sperm that fertilize an egg. So here's a haploid, and a haploid. This is a normal thing that happens in flowering plants, and it's probably one of their biggest success evolutionary steps. That is what the delicious part of a seed actually is. It's a triploid bit of developing plant. Allopolyploidy. Allo means foreign or away, separate. I always remembered it because if you were separated from someone and they were like really far away, you'd have to be like, allo. Allo is separate. Allopolyploidy is where you get one set of chromosomes from one parent and a different number from another parent. It's probably from a different species. In animals, this results almost exclusively, as far as I know, exclusively in either a not viable offspring or a sterile offspring. The mule is an example of something that is resulting from allopolyploidy. A horse and a donkey have different number of chromosomes. They can make a baby together, that's a mule, but that mule actually ends up having an uneven number of chromosomes and can't go through meiosis. It's often allopolyploidy as a consequence of hybridization, and that just means putting two species together. And I'm sure you did a bunch in Bio 1, but the mule and from the donkey and the horses seems to be everybody's favorite. In plants, this is not something that leads to sterility, and it's something we encourage in many ways to cause offspring that are maybe produce more fruit or more sugar or grow taller or have more flowers than their parents because they have a different number of chromosomes. And each one of those chromosomes now has another gene for making flowers or sweetness. There's not a dosage error where it makes the plant sick. It actually makes the plant look more robust in some cases. And in rice, this is actually how we breed rice with lots of grains. We do find if you have to keep back crossing them though. If you keep crossing two allopolyploids together, you end up with a problem after a while in non-viable offspring. An autotetraploid organism are when this is auto is self, tetra is 4, so this is an auto 4N. The chromosomes replicate during S phase, but the cell doesn't divide. This means that we double the number of chromosomes, but then don't take the cell apart. So we go through prophase, you can see they form sisters. We go through late prophase into prometaphase, but then they never form the spindle fibers to pull them apart. They separate the sisters from each other. Oops, that was supposed to be a straight line. They separate the sisters from each other. And now this cell that started out as 2N equals 4 is now 1, 2, 3, 4, 5, 6, 7, 8. It has 8 chromosomes inside of it. I made this red because this might be of interest to you. I'm not really going to ask you big questions about it, but just so it stands out. Colchicine, I don't know how to say it. Colchicine, I can read it but not say it. This is a medication to treat gout that we use in humans all the time. Gout sounds like it's made up, but it's not. It's a real disease that has some real consequences where people build up uric acid in their joints. It's very painful. But it also turns out if we expose plants to this chemical, it causes them to become autotetraploids. It interferes in their meiosis. And we do this to cause them to mutate. And then we create varieties of plants that are covered in more flowers or have more expressions of genes that we actually like. Most of our modern plants that we eat or that we sell, or the ones you buy, the flowers at the garden, and they have the purple planter underneath them, and I can't remember, they're like miracle plants or something. And you have, when you break the little plastic off, it says you're not allowed to propagate them. That's because they've been modified not through insertion of genes, but actually by exposure to chemicals or radiation that causes them to mutate the number of chromosomes. We cause them to be autotetraploids. Allopolyploidy is often the result of hybridizing two species that are closely related. I mentioned it before, but I wanted to put it here with all of the vocab words. It means that we can hybridize something, but often that plant is sterile. This is great. If you're producing a plant that you're selling to gardeners, you kind of want it to be sterile. It's also great if you're producing a plant that would be non-native to a region, and I don't want it to propagate and spread into my region and become invasive. I can put something pretty in my garden without all the consequences of it reproducing and out-competing some of my native species. Highlight a little bit here. Allo means different, auto means self. Poly means many, ploid is sets of chromosomes. When I have the questions on the homework, I think it's not bad to just have this set out in front of you in your notes so you don't muddle all of these words together. After you see them a couple times, they'll start to separate. Their concepts will separate for you. I made this last one. I guess I have two more. Endopolyploidy, I think you might be interested in this. I didn't make it red. I don't have any questions about it on the exam, but I think it's something you might be keen on. In some diploid organisms, including you, we see cells that are polyploid under normal conditions. The chromosomes are replicating, but they're not going through mitosis. This is something we see in your liver, and we think it might have something to do with the amazing regenerative ability of the liver. I pulled some papers if this is something you're interested in. This is a little bit thick to read, but it links back to a bunch of other resources on our understanding of liver replacements. So this is hepatectomy. This is removing part of the liver, and then looking at the growth and development of that liver as it ties back to endopolyploidy. The cells inside, endo is inside, become polyploid. This is also found in cancer cells, and it's one of the hallmarks of cancer cells. They're going through mitosis inappropriately. So they're sending the signal to go through S phase, entering past the checkpoints, which they should not be able to pass, and now they have too many chromosomes. And I'll find tumors of cancerous cells that are not only making poisons because they're making too much of genes that are dosage dependent, but now they're also going through mitosis in completely random order and without any checkpoints. Finally, there's one more. Yay! Allotetraploid is crazy and just in plants. I don't know. I'm for sure no animals are involved in this. And if you want outside of the scope of this class, but it has to do with the segmentation in animals and our developmental process, it's really important that our heads are on top. It's really important that animals come together and construct their bodies during development in the same way. Plants really aren't under that. They can put like a flower over here or a flower over here, and it's all good with plants. So they're a little bit more robust to chromosomal mutations and abnormalities. An allotetrapod means that we have 4 different genomes involved. So 4 different contributors of chromosomes. They may be the same numbers, they may be different numbers, but the plant is just trucking along doing that. And this is cotton. Cotton does this. And so one of the reasons that there's so much, this is where cotton comes from. There's so much cotton on a cotton plant is it actually has ancestors from multiple species. An amphidiploid is where we have an allotetraploid where we know both parents. This cotton is an allotetraploid and it's amphidiploid. I know where all 4 parents come from. So just kind of crazy to think about. And we do think in the studies of looking at plants that this is their major mechanism for evolution. Whereas in animals, it's more like natural selection that drives adaptation. In plants, it looks like it's crazy chromosome problems. And since they can survive that means that they just get something new and exciting. I have some practice with me. I included these in the PDF that you have, but I didn't include the answers. I do have the answers here, so I was going to do them and then give you the answers in the video. If you are not sure where the answers come from, let's practice them in class as well. I want to, and these are very similar to what I will give you on the next exam. The next exam is not cumulative, so it doesn't go back and talk about some of the stuff we directly have covered for the first exam, but we can't forget about meiosis, right? Or mapping or all those other things. So I want to keep tying you back without directly asking you those questions. A horse is 2n equals 64 normally, and a donkey is 2n equals 62. This donkey and this horse both make gametes that are haploid. So a horse makes a n equals 32 gamete, and a donkey makes an n equals 31 gamete. Those are different numbers. And although in many cases, if I hybridize 2 species together, this would interfere in the development of the offspring, it does not interfere in the development of a mule. And a mule is a hybrid of a horse and a donkey. A mule then has this, let's say, this better be the sperm. Donkeys are much smaller than horses, and this is the egg. How many chromosomes are in a diploid mule, and what does that mean? So I'll give you a chance to pause me or work through it, and then I'll come back and answer the question. So here the answer. Donkeys have a true ploidy number. They're diploid, but their chromosome number is 63. This is why they're sterile and they can't pass on their genes to the next generation. They generally, how do you go through meiosis with this? It's an uneven number. A human individual, question number 2. A human individual with 47 chromosomes has a monosomy, a trisomy, a monoploidy, a diploidy, or a triploidy. And as you're thinking about this and trying to answer it, I think you should define all of these on this part of the slides if you're printing them out or in your notebook, because these words, they sure sound the same. Okay, I'm going to skip ahead to the answer. A trisomy, they have 1 extra, so this is 46 plus 1. This person maybe has Down syndrome or Patau syndrome or Klinefelter syndrome. They have 1 extra chromosome. This guy has Klinefelter syndrome. He has normal color vision. His mom was colorblind, but his father had normal color vision. So let's make his mom and his dad. I'll give you, if you want to pause me and you do it on your own, and then you can unpause me and I'll solve it. I'm back. His father is XY, and we'll just make a mark. His father has normal color vision, and his mom was colorblind. Let's give her, that means that she must be carrying 2 different alleles for colorblindness. Colorblindness is recessive. Can you tell, because he has normal color vision, can you tell where he got that extra X from? So Klinefelter syndrome, he must have X, X, Y. He can see color. So his combination must be color vision, no color vision, because mom had to give him something, and a Y. That suggests to me that dad gave him an X and a Y. He can see color, even though his mom is colorblind. Here, I'll put it back so he can see. And it's father. With that, that's the end of this video. I know these are a little bit shorter. I do have a quiz and homework. You're watching 7 and 8. There is one more little video here where we're going to talk about some things that happen when the chromosomes get messed up. Not the number, but the chromosomes themselves. That's part 2 of this video. So I'll post it up separately, and I'll see you in lab and in the next video. Right. Bye-bye.